Couple filtering method is able to identify the identical speckles and reduce the feature motion decorrelation. Compared to other speckle tracking method, couple filtering method is the best to minimize the influence of the tissue deformation, and it can handle almost all kinds of the tissue deformation, including the 2D and 3D. However, the limitation of couple filtering method is a long computation time. Therefore, we have this project to rebuild the couple filtering program and accelerate it. Before we go into the program, let's talk about how the couple filtering method works. First of all, we take the image of the tissue before deformation and the image of the tissue after deformation. Let's look at the equation A on the top right-hand corner. The speckle of the tissue will be represented by the variable I, in which x is a position vector and p is a refractive coefficient. We can see the speckle before deformation is equal to the convolution of the tissue scatter and the point spread function, also known as the PSF. By a special procedure called the affine wrapping method, we can get the equation B, showing that the speckles after the deformation is equal to the convolution of the tissue scatter and the PSF expanded by the coefficient m. Looking at the equation A and equation B, we can find that the pair of ultrasound images will share the same tissue scatter. That means we can return the speckle by making the speckle before equal to the speckles after. After that, the remaining difference between the speckle before and the speckle after lies in the PSF term. We can see one of it is expanded by the coefficient m and the other is not. Then the filters will be applied to the speckle before and the speckle after. By comparing the two equations, we can see that the speckle patterns become identical to each other. The procedure of making the speckles before equals to the speckles after is called the couple filtering method. Inside the equation, there are two variables, the m and t. You must be curious, what are they means? M is a 2 × 2 matrix describing the tissue deformation and the rotation in the local area. And T is a 2 × 1 factor denoting the translation of the tissue scatters. Inside the couple filtering method, the estimation of the motion parameter is achieved by searching for the optimal motion parameters. This procedure, we call it the search for the motion parameters. The correlation coefficient C between the speckles before and the speckles after is maximized, and this coefficient C is the ultimate goal of our program. Here is our program of the couple filtering method. You will see the formation of PSF and the command of linear convolution is done.
Next, we're going to talk about the implementation of the couple filtering method for analyzing the ultrasound image with deformation. It will be separated into 3 parts for explanation. First, we're going to show you guys the searching algorithm used by Tenzo, and then we're going to show the correlation matching process and talk about the cost-defined strategy. Here is the main algorithm proposed by Tenzo in his thesis used for the search of optimal motion parameter. Please take a look. Next, we're going to mainly focus on the explanation of correlation matching, aka the implementation of couple filtering method. About the correlation matching, first we're going to apply the couple filtering method on both images, pre-deformation and post-deformation. Next, for post-deformation image, the application of affine wrapping method is needed for the compensation of deformation. Next, we're going to obtain the resultant image from the 2 applications of couple filtering method respectively. Last but not least, we're going to calculate the correlation coefficient between the 2 selected windows from the 2 compensated images correspondingly to check the optimality of the used parameter for that specific location. However, the problem is correlation matching can only be performed for one set of correlation windows at once. Then how can we be able to obtain the search of optimal motion parameter for every single piece of local area of the image? Cost-to-find strategy can solve the problem. First of all, a sphere grants of correlation windows are used at cross scales, where dense grants are used at fine scale. Center of the correlation window form a square lattice at each scale, where the number of correlation windows along each direction, vertical and horizontal, is roughly doubled at each final scale. The search range of motion parameters is reduced by half at each final scale, and a refined search is performed in the local neighborhood of the interpolated estimation result from the closest scale. And here is the actual program that we have rebuilt according to the thesis proposed by Tenzo about the application of the cost-to-find strategy and the correlation matching process. Please feel free to take a look. And here is the estimation result. Left-hand side, the result of the search in scale 1. All 12 windows have been assigned their corresponding optimal motion parameter in different directions under different layers of the multi-dimensional array. After several scales of stretching, we can reform the image from the data of the 24,897 windows. We can see that compared to a direct correlation method, couple filtering method can obviously reform a much clearer image to describe the different type of deformation of the shifted region. So right now we're going to talk about the performance improvement. There are a total of 3 approaches in programming practice modification of MATLAB in order to further accelerate the couple filtering method functions. They are the matrix pre-allocations, convolution in frequency domain, and the last step further factorization. The speed tests for the couple filtering method shown in here are only for the pre-deformation. First one is the matrix pre-allocation. We can first relocate the matrix in advance instead of allocate them inside the function or loops. In other words, we can create a matrix and define the corresponding size before the computation. For example, in this function, we can first relocate a 1001 times 201 size matrix for the formations of the point split function hm during the later computation inside the last loop. Next one is the convolution in frequency domain. By using the convolution property in Fourier transform, we can transfer convolution between the image and the PSF function in spatial domain into a multiplication in frequency domain by using the internal functions of fast Fourier transform FFT inside MATLAB. By comparing the two results, we can figure out that performing a convolution in frequency domain can greatly reduce the execution time. Last but not least is last step further factorization. In fact, matrix calculation can be used as a replacement of the last loop implementation for matrix assignment. The execution time will be reduced because of the strong mathematical calculation ability of MATLAB. By using the internal function matchgrad, we can create two matrices that have an identical size with the point split function for replacing the two layers of the last loop in the original code.